Good afternoon. Welcome back. This is part 2, B cells and T cells in adaptive immunity. I just want to go over some similarities and differences. You're going to make a table from this. I'd like you to compare and contrast B cells and T cells. So I kind of played it out for you. Both of these are lymphocytes. They primarily move around in the lymph. They are generated in the bone marrow, but B cells mature in the bone marrow. T cells are going to mature in the thymus. Another difference is the way they look and the way the protein receptors on the outside of these cells look. B cells have a Y-shaped receptor. This is a protein. I have several different versions of it here. This is a cartoony version. This is a zoomed in version looking at just the Y shape here. Here's the Y. This purple and green, so this whole part, if you're colorblind, is showing you the protein that's sticking off of the B cells. This part, when it's stuck to the B cell, is an antigen receptor. It is looking for an antibody generator. When it's recognized and binds to the antigen, this B cell will make lots of these Ys and they will break off into the blood. These Ys become antibodies. B cells target the pathogens in the blood, so things that are moving from one cell to the other and moving around in your body. They have this variable region here, and there's another variable region here, or the tips of the Ys in these pictures. These are randomly generated protein shapes. They do this by generating random sequences of mRNA in one small piece, generating random proteins. This way they can come up with a lot of possible solutions for binding with anything that might pop into your blood. When a B cell is activated, because it does wait until it's needed, so they're not all going around all the time proliferating and making lots of white blood cells. You would be responding as if you were sick all the time. But when a B cell is activated, it detects an antigen. Whoop, 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 and the B cell binds to the antigen. When it binds to the antigen with its antigen receptor, it's right here, binding to a pathogen, and the epitope of a pathogen, the B cell's antigen, Y-shaped antigen receptors are binding to this bad guy. We'll make him mad. When this binds, this stimulates a response, which I'll go into in a next video a little bit more detailed. This stimulates a response that causes the proliferation or lots of copying of this B cells, and even more importantly, many antibodies of this particular shape are generated. These are proteins. They break off of the cells. They're immunoglobulins that move around inside of your bloodstream, and they fight off anything moving in your bloodstream. Again, this is humoral, which means it takes place in your body's humors. This is a really old term. Your humors doctors used to believe a couple hundred years ago that people were made up of 4 fluids: the blood, the black bile, green bile, and mucus. And I mean, you can see why it feels like that. Some days it feels like that's all we are. But your humors could be needed to be in balance for you to function properly, way before we knew about how the body worked. This word gets held over here. A humoral response means in the blood. Old word, very new understanding of how it works. T cells, I want you to compare and contrast them. Remember, they are also lymphocytes, but they're not Y-shaped. They do not make antibodies, but they do play a big role in your immune system. Remember, they target your cells. Now, we've talked about a couple not specific things that attack your cells that are yours, like natural killer cells that non-specifically kill your cells. These target your infected cells, but they are specific. Now, they have some similarities to the B cells. They have these proteins that stick off the outside. They are not Y-shaped, they are line-shaped. And they still have this antigen binding site. Same role as in the B cells. These are randomly generated. You see, it's called a variable region. This randomly generated area has a randomly shaped protein that binds with randomly shaped things that your cell is showing that it is making. If your cell is making something that is not you, so it's making a virus protein, it's making something that's very wrong, your T cells can bind with it and trigger a cascade of responses that are going to destroy your sick cells. So I want to redefine this word because it's one of the words that tricks people up a lot. Antigen is an antibody generator. Antigen. But it means that it's an epitope on a pathogen or something that doesn't belong. I would write something wrong. A protein, or let's go with protein, it's easier. A protein that is wrong. So T cells, remember, we'll keep reviewing, are these cells that have a just straight line. They are not going to generate antibodies. They're going to generate a response that kills your infected cells. So they are looking for cells that are you, displaying something that is not you. Antibody generators, or antigen fragments, can be displayed by cells that are infected. So these are your sick cells that are making proteins that don't belong. Or they can be displayed by cells who are phagocytic, cells that are involved in their innate immune cells. They ate the bacteria that infected you, and as they ate it, they displayed a display of what did I just eat. Those phagocytic cells that are eating foreign invaders into your body display what they are doing. Like all cells in your body, they're showing the rest of your body, important part of being multicellular, what everyone's doing right now. A phagocytic cell eats and displays a piece of what it just ate. As soon as it eats, or phagocytizes, it turns into an antigen presenting cell. It's displaying an antigen or something that's going to stimulate an antibody response. And we'll call it an APC. I'm going to keep saying the whole name because I think there's a lot of vocab in this lecture, and it's right toward the end of the semester, so things can get a little overwhelming. The phagocytic cell eats something that doesn't belong, displays it, and as soon as it displays it, it presents it to the world. It becomes an antigen presenting cell. And that's something that a T cell can bind with and say, hey, I recognize this as being bad. I'm going to leave you here. Create a table, B cell versus T cell. I know we have not gone into details yet. See if you can just get them straight. Here is a real B cell. This is an actual picture, so you can see they really don't have proteins that you can look at, but you can see they're a fairly complicated cell. Ooh, look, here's even a mitochondria in one of them. How cute. It's a cute one. Two of these are correct. Two of these are not correct. I'd like you to be able to identify the answer and come back with the answer before the next video. And I'll see you in the next video. Put it all together. All right. Bye-bye.